Hello, my name is Sia Garg, and I'm going to be doing my presentation. Today is July 21st, 2024. Okay. So I'm going to be talking about the role of the pancreas and liver in regulating digestion. So first I'm going to be talking about the pancreas. So the pancreas, as you can see here, is located very close to the main digestive tract. It's very close to the stomach and the duodenum, where it does the primary of its work. So the pancreas has both endocrine and exocrine roles. We're going to be talking mainly about the exocrine functions, which are used in digestion. However, it is important to acknowledge the endocrine functions. So this allows for the maintenance of blood sugar levels. And it does it through the release of peptide hormones such as insulin, glucagon, and somatostatin. So the pancreas is mainly made out of acinar cells, and these are the cells which produce the pancreatic juices. So the pancreatic juice is a bicarbonate-rich alkaline secretion, which means that it's mainly basic, which contains digestive enzymes that can be used within the duodenum, which is in the small intestine. So in this slide, I'm going to be mainly talking about the hormones within the pancreatic juices and the enzymes. So these enzymes essentially work on biomolecules, so lipids, carbohydrates, and proteins. The only biomolecule it doesn't directly work on is nucleic acids. So pancreatic amylase, it breaks down large carbohydrate polysaccharides into small disaccharides. So as you may know, there is amylase within your saliva, which is in the mouth. However, and this does break down initially the carbohydrates. However, like a lot of times, people when they eat, they don't chew for that long, or your food doesn't stay in your mouth for that long. So while that amylase does break down some of the carbohydrates, it's not really able to focus on breaking it down. So the pancreatic amylase is really able to do that, as your food stays in your small intestine for a little bit longer than in your mouth. So pancreatic peptidases, there's multiple of them. There's trypsinogen, chymotrypsinogen, and carboxypeptidases A and B. So trypsinogen and chymotrypsinogen are the zymogen forms of the enzyme. The active forms are called trypsin and then chymotrypsin. So initially, these peptidases will be released in their zymogen form. However, they have to be cleaved in order to be used in their active form. So enteropeptidase is something found within the brush borders of the duodenum, and this can work on the trypsinogen to cleave it and make it trypsin, and then the trypsin can later on activate the other zymogens. A fun fact to know is that the carboxypeptidases A and B, they're initially called pro-carboxypeptidases A and B, and trypsin can work on those as well. And then pancreatic lipase, this breaks down the fats into free fatty acids and glycerol. Yeah. So just like to really hone in on the connection of the pancreas with the digestive tract, the pancreatic juices will be transferred to the duodenum through a duct system. As you can see here, the duct system is in the middle of the pancreas, and it kind of just like runs down the middle. So the acinar cells will secrete the juice directly into the ducts, and then the ducts will empty into the major and minor duodenal papillae, which are located in the duodenum. So if you like really look here, these two holes, this is the major and this is the minor duodenum papillae, and they're just holes that allow the juice to go into the duodenum and allow the enzymes to work on the compounds once they get into the duodenum. So now I'm going to be talking about the liver. So the liver is located in the upper right quadrant of the abdomen. So as you can see here, it's right here, and it contains bile ducts, which connect both to the gallbladder, which is another accessory organ, part of the digestive system, and then the small intestine, which I talked about before. So one of the liver's main functions is to produce bile. And I'm going to be talking about this in the next slide further on. But bile can be stored in the gallbladder if it's not needed right away, or it can be secreted to the small intestine and like undergo its function essentially. So the liver can receive blood, which is drained from the abdomen portion of the digestive tract through the hepatic portal vein system, and it's essentially gonna take this blood and process it, and then later on, once it's processed, this blood can go to the super vena cava and then to the right atrium, and then go through the heart essentially. And a really big important process of the liver is to essentially maintain blood glucose levels similar to the pancreas. So what it does is if there's a lot of glucose, it will take up the excess glucose into glycogen, which is the storage form of glucose. And then if there's less glucose, so when you're fasting or you're starving, then it's going to produce glucose through glycogenolysis, which is the breakdown of glycogen, or gluconeogenesis, which is kind of the opposite process of glycolysis. There's just steps here and there that are slightly different. And then it can mobilize fats into lipoproteins, which is pretty important. And then it can also detoxify endogenous and exogenous compounds. So endogenous is made within the body, exogenous comes from outside the body. So an example I used was converting ammonia, which is a very toxic compound, into the urea, which can be excreted from the kidneys. And then a very important part is detoxifying and metabolizing alcohols and drugs and medications. This is very important because if this is not done, then the alcohol and drugs are very harmful and detrimental to your body. So just to hone in on the why the liver is so essential to the digestive system, and this is mainly because of the bile production, which I had talked a little bit about before. So just to go into it, bile is composed of bile salts, pigment, and cholesterol. Cholesterol is a type of steroid. It's a precursor to it. So the type of pigment is bilirubin. And bilirubin is something that's made from the breakdown of hemoglobin. So once hemoglobin is broken down, the bilirubin will go to the liver. It's going to attach to a protein, making it conjugated, and then be secreted to the bile, which will later be excreted. Something to know is that bile is amphipathic, so it has both polar and non-polar aspects to it, allowing it to interact with both types of environments. And its main function is to emulsify the fat. So emulsification is essentially bringing the fat together. It does not mean that it's breaking down the fat like pancreatic lipase does. Emulsification and breaking down are two very different things. And then something to note is that it can synthesize proteins. So albumin is a protein that it can synthesize, which maintains plasma oncotic pressure and can also work to carry drugs and hormones throughout the body. And then it can also synthesize clotting factors, which are used in blood coagulation. So if you get hurt, you start bleeding, you get a scratch, it's the clotting factors which allow for the blood to stop coming out and for it to be clotted. So these are just like all the important aspects of the liver and the pancreas. So I have made a worksheet that can be used to really understand the functions of the pancreas and liver, and then there's also an answer key to it. But thank you.